Assalamualaikum and good day. I'm IFFAH Binti LAILI, student of Faculty Health Science from Universiti Kebangsaan, Malaysia. I'll present to you my research study entitled Chloroquine-Induced Lysosomal Dysfunction, Increased Amyloid-Beta Expression in Human Brain Endothelial Cells. To begin with, decline in cognitive brain function represents one of the main health challenges of aging. This is known as neurodegenerative disease. Alzheimer's disease is one of the most common type of the disease that affect older population age 65 and above. According to Alzheimer's Association, about 50 million people suffer from Alzheimer's disease worldwide and 10 million new cases are recorded each year. The Alzheimer's disease is characterized by progression of symptoms that hinder the performance of daily activities. These include memory loss, impaired communication, difficulty in walking and swallowing, as well as changes in behavior. Neurodegenerative diseases are related to the deposition of protein aggregates, like in Alzheimer's disease, the deposition of amyloid beta peptide that are generated from the sequential cleavage of amyloid precursor protein by beta secretase and gamma secretase in the brain is known as the main neuropathological hallmark. Deficits in amyloid beta clearance plays pivotal role in Alzheimer's disease progression, and its aggregation lead to the disruption of brain endothelium that contribute to the vascular dysfunction. Lysosome is the primary degradative organelle in cell that function to degrade any macromolecules and organelles that are damaged or aging. It is known as the terminal nexus for crucial autophagy degradative pathway in cell. The lysosome degradation system is important in maintaining cell homeostasis. Its dysfunction will negatively affect the autophagy degradation pathway, and the impairment in lysosome degradation system has been said as one of the primary contributing factors that cause deposition of amyloid beta aggregate in the brain. However, it is still not clear if lysosomal dysfunction can affect the brain endothelium in terms of amyloid beta expression in Alzheimer's disease. Therefore, the aim of this study is to observe the effect of lysosome dysfunction on amyloid beta expression in human brain endothelial cells. In this study, chloroquine, which is a commonly used lysosome inhibitor, was utilized to treat human brain endothelial cells. Chloroquine is a weak-base compound that will accumulate in acidic compartments such as lysosome and will cause alteration in lysosome pH. Chloroquine-induced lysosome dysfunction will lead to blocking fusion between lysosome and autophagy vesicle, or also known as autophagosome, and thus will impair the degradation system of autophagy. The method of this study involved culturing of human brain endothelial cells in pre-coated flasks with 0.1% gelatin, followed by MTT assay to examine the cytotoxic effect of chloroquine on HBEC5I cells, as well as to establish dose. The inverted light microscope was used to observe cell morphological changes after treatment with chloroquine for 24 hours. The automated microcapillary immunoassay was conducted to confirm the lysosome dysfunction by measuring lysosome and autophagy markers, which are LAM1 and LC32. Enzyme-linked immunosorbent assay was conducted to measure the expression of amyloid beta 42 in cell lysate and supernatant. And for the statistical analysis was conducted using GraphPad Prism. The sample were prepared in triplicate and the data were presented as mean plus minus standard deviation. Based on the MTT assay result, chloroquine is a dose-dependent compound toward HBEC5I cells, which shows cytotoxic effect increases with increasing chloroquine concentration. At 12.5 micromolar, the viability of cells started to decrease and was significantly reduced at 200 micromolar. The IC50 value was obtained at 125 micromolar. In order to establish dose, an optimal chloroquine concentration that did not affect cell viability is needed. Therefore, IC10 value, which is 17.5 micromolar, and IC25 value, which is 70.5 micromolar, were obtained and performed along with 125 micromolar in the subsequent experiment at similar time point of 24 hours. Cell morphology was observed after 24 hours treatment with chloroquine. At low concentration of 17.5 micromolar, there were no morphological changes observed. Meanwhile, at 70.5 micromolar and 125 micromolar, clear vacuole formation in cytoplasm were observed, as well as cell shrinkage that were more prominent at highest concentration of 125 micromolar. Lysosomal-associated membrane protein 1 is major component of lysosome membrane. Hence, it is an appropriate marker to measure the lysosome integrity. Based on the densitometry analysis, band intensity for LAM1 expression was observed at all concentration with molecular weight of 180 kilodalton. The LAM1 expression showed decreasing pattern as the chloroquine concentration increases. Results obtained indicate that chloroquine exposure led to the disturbance of lysosome integrity, which is also known as lysosomal membrane permeabilization. Lysosome dysfunction is related with the accumulation of autophagosome in cell. Therefore, microtubule-associated protein 2, which is the autophagosome marker, was measured in order to confirm the lysosomal function inhibition by chloroquine. Based on the densitometry analysis, band intensity for LC32 expression was observed at all concentrations with molecular weight of 18 kilodalton. The result obtained showed increase as the chloroquine concentration increased. Increased amount of LC32 expression correlates with the accumulation of autophagosome in cell, which is due to the chloroquine activity by blocking the fusion between lysosome and autophagosome. Amyloid-Beta 42 expression was measured in two conditions, which is in cell lysate represent the intracellular expression and in supernatant represent the extracellular expression. The expressed amyloid-beta 42 protein was increased at all concentration in both conditions. Meanwhile, in lysate, 70.5 micromolar showed significant increase as compared to the control. The higher expression of amyloid-beta 42 was observed in cell lysate as compared to the supernatant. This is related with the accumulated autophagosome, which is known as the site for the APP metabolism. In conclusion, chloroquine successfully inhibits the lysosome function observed through the decrease in LAM1 expression due to lysosomal membrane permeabilization and increase in LC32 expression, which due to the accumulation of autophagosome in cell. The effect of chloroquine-induced lysosome dysfunction caused increment in amyloid-beta 42 expression in both intracellular and extracellular, but more prominent in intracellular. Further study need to be conducted to examine the effect of lysosome dysfunction on the permeability of the brain endothelial cells. This study was funded by Ministry of Higher Education Fundamental Research Grant Scheme. That's all from me. Thank you.